Hello, and thanks for coming to this talk. I'm going to tell you about a project that we've done on the genus Reynoutria, the Japanese knotweeds and relatives. This is a group that are invasive species in North America and Europe. They're native to Eastern Asia, and there are three taxonomic entities, two species, and they're hybrids. So the species are giant knotweed Reynoutria sakalensis, Japanese knotweed Reynoutria japonica, and the Bohemian knotweed known as Reynoutria bohemica. And these all have a very similar introduction history where they've been brought to Europe and North America, where they've proliferated quite a bit and almost equally on the same time scale. This is their relatively small native range, and they are a relatively small genus in this larger family Polygonaceae. And depending on who you look at for a source, you might find these classified in a different genus. They've been classified variously in Polygonum or Fallopia, and based on this phylogeny, they are nestled pretty firmly now in the genus Reynoutria. So there has been a good amount of investigation of the species in Eastern Asia, and showing pretty cleanly, based on the plastid sequences, a big block of the Japanese knotweed in this blue color, a big block of the giant knotweed in this brown color, along with a couple of accessions that they sampled in their study from Russia, United States, and United Kingdom. These are outside of the native range for those species. So pretty clean phylogeny for the plastid data on the left. The nuclear data a little more exciting, I guess I'll say, where we have a big block of sakalensis, giant knotweed type, and in brown again, and blue for japonica. And seeing a couple of instances, this one from Russia in the japonica group, having actually two separate sequences, both falling within Reynoutria japonica clade. This individual from United Kingdom, two sequences, two kinds of sequences of leafy for the sakalensis type. And then this is a species, an individual from United States. So some evidence of multiple combinations of leafy sequence, but not too widespread here. This one individual with a mix of japonica type and also sakalensis type from Honshu, Japan. So we were interested to get a little more data on these kinds of sequences from plants in North America. And what we got is shown here. This is our plastid data from only the MAT-K gene. So pretty cleanly, again, showing individuals having either a japonica type or a sakalensis type MAT-K sequence. And all of our individuals were identical if they had the japonica type, or identical if they had the sakalensis type. So not a lot of variation seen there. In the leafy gene, on the other hand, lots of evidence of multiple copies and crossing between individuals having these multiple copies. So any one individual shown in these solid lines, one individual can have a mix of sakalensis type, japonica type, and multiples of each of those even. So certainly not every individual that we sampled, even in our small geographic sample of Wisconsin, we found two kinds of sakalensis sequence, four kinds of japonica sequence, and all kinds of combinations between. So this is largely attributable to polyploidy. Having these multiple copies of genes is consistent with what has been observed to know that these plants are polyploid. They've got tetraploid, hexaploid, octaploid chromosome combinations. And we're seeing, as a result of hybridization, lots of combination possibilities here. So these are a graphic demonstration of the hybrid gene combinations that we've seen. The top part of the shape is the maternal plastid sequence, so lots of japonica plastid contribution, and a couple of individuals with this brown color sakalensis plastid contribution. The squares are sequences we didn't observe singly in individuals, but potentially coming from one or more parents. And then the plants we did observe, we observed some, as this one, with a purely japonica sequence, even though it has three kinds of japonica leafy sequence, or this one that has purely sakalensis sequence. But we also found the majority of individuals were some form of hybrid, so some combination of japonica and sakalensis in the same individual with a variety of different parentage. So S2 for Sacchalinensis or S1 for Sacchalinensis. So as a result of what we found, we were able to develop a diagnostic PCR that can amplify the Japonica type or the Sacchalinensis type. You can see some individuals had only Sacchalinensis type, some had only Japonica type, and many individuals here, the columns are individuals, many individuals had one of each, as we saw in the pie charts there. So just graphically through Wisconsin, a little bit of distribution, geographic patterning where the Sacchalinensis individuals are found more in the north. The pure Japonica individuals are found in the northwest. One kind of hybrid predominates in the southeast. Another kind of hybrid predominates in the northwest. So a little bit of a pattern there that might be consistent with people moving these around or genetic combinations that are not breeding more widely.